So I'd just like to give an overview of the agenda and what the presentation will be on today. So as mentioned, I'll give a very brief overview to Eurofins Discovery for those people who aren't familiar with the company. We'll also have a look at what is ADME, and then from there we'll look at some of the essential early ADME assays. So these are solubility, protein binding, metabolic stability, absorption and permeability assays, and also lipophilicity. I'll also spend some time looking at some of the later stage ADME assays, introduce you to an ADME screening cascade. ADME should be tailored depending on the therapeutic area that you're working on, have a look at some case studies, and then do a brief summary. So for those people who are unfamiliar with Eurofins Discovery, we're a network of a number of laboratories worldwide, and we focus on early drug discovery. So we can take people from very early hit finding, target validation, all the way through the drug discovery process to identifying a preclinical candidate. So we do this by combining a number of different disciplines. So we start with chemistry, we have in vitro pharmacology, we have one of the largest selection of off-the-shelf target-based binding and functional assays. I think we're probably best known as an organisation for our safety pharmacology. Obviously, we have ADME tox services, which is what I'll be talking to you about today. One of our flagship services is our translational phenotypic platforms. Then we have in vivo, and then finally we have custom assays and products. And then we can bring all of these together into fully integrated drug discovery programs, which we call Discovery One. So we really do offer a very comprehensive selection of assays, support, chemistry to be able to take your program essentially from a concept, from an idea, all the way to preclinical development. Just to touch on where we're based. So as I mentioned, we're a network of 9 laboratories. We have a very much global footprint. So we have sites within North America, within Europe, in the UK, France, and in Spain, and then also in the Asia region in Taipei. The reason for highlighting this slide is to also show you where we have our ADME tox capabilities. So we have those within a number of sites. So in St. Charles in Missouri, that's where our Centre of Excellence is for ADME. We also have ADME tox capabilities in Poitiers in France. And then finally, we're also establishing them at our chemistry site in Spain. So this is very important for those people who are really looking for short turnaround on their make test cycles, having chemistry right next to ADME. So that's just to give you a really quick overview of Eurofins Discovery. I understand that my colleagues, Hannah and Makoto, are online as well. If you have any questions regarding any of our services, please don't hesitate to contact them. So now I'm going to move on to talk about ADME. So what is ADME? So this is a very, very quick overview. So I have to say that within the hour, it will be very much a high level introduction to ADME. That it's a very complex discipline within drug discovery. It's become more and more recognised and vital in recent years. So I said, what is ADME? So ADME stands for absorption, distribution, metabolism, and excretion. So absorption is the ability for a compound to pass through barriers such as the intestine or nasal lining, lungs, or skin. For distribution, we're talking about the ability of a compound, of how it's metabolized within a system. Metabolism is how a compound is broken down. It's normally focused on the liver, so 60% of metabolism is performed by the liver. And then we also need to look at key factors such as drug-drug interactions and effects on metabolites. These are particularly called out by organisations such as the FDA and the CTA. So drug-drug interactions and metabolite identification are really key if you're doing any type of new drug submission to one of the regulatory bodies. And then finally, excretion is how the compound exits the body. So some of the early ADME testing. So early ADME is essentially a balance between all of the key properties. So companies often begin with solubility, and that's looking at the compound solution properties in various different types of solution. So solubility can often be the first assay that you look at just on its own, and then you'd move on to some of the different assays. So metabolic stability, which is the biotransformation of a compound within the body. Then protein binding, which is binding of the therapeutic compounds to plasma or serum proteins. Lipophilicity, which is the affinity of a compound to bind within a lipid environment. And then finally, permeability, which looks, as we mentioned before, a permeability of compounds across different cell membranes, such as the intestine or the nasal lining. So all of these things are balanced to try and develop small molecules that have really optimal drug-like properties and which will increase the chances of success of your compounds progressing to the clinic. So now I'm going to go into more detail on each of these different early ADME assays. So looking at compound solubility. So compounds with insufficient solubility are known to carry a much higher risk of failure during discovery and development. This is because insufficient solubility may compromise other property assays, can mask additional undesirable properties, and influence both pharmacokinetic and pharmacodynamic properties of the compound. And all of these may finally affect the developability of the compound. So solubility is really important, because if you're trying to assay insoluble compounds within any other different type of efficacy assay, it could be that you're not getting correct results because of this compound not solubilizing. It may be that you're not getting the full compound effect within your efficacy assays. Compound solubility also has a direct impact on other ADME properties of the drug. So insoluble compounds often have lower bioavailability, which again can make them very difficult to dose as you move forward into preclinical studies. They're highly bound to plasma proteins. As I mentioned, they also tend to precipitate out of solution when conducting in vitro assays. There are some strategies that you can adopt to counteract insoluble compounds. So you could look for novel formulation strategies. However, these can be very costly and may lead to delays within your preclinical development programs. As I mentioned, it is recommended to address solubility issues early on. So as an anecdote, I've worked with organisations who will screen their compounds within solubility assays as the very first assay. If those compounds do not reach a predefined solubility threshold, they will not be tested in any further assays. This means that the chemists then have to go back and re-optimize the compounds. This can be a very effective strategy in making sure that anything that you take through to the clinic has sufficient bioavailability. So some factors affecting solubility include the molecular weight of the compound, the lipophilicity, which we'll talk about later on, particle size, solid and salt forms of the compound, and also their pKa. So there are different assay formats for measuring solubility. So kinetic solubility tends to be what you use at an early stage. So at the really early discovery, and then thermodynamic solubility is a much more intrinsic assay, which you would use for later stages, more towards a preclinical candidate selection phase. Just to give an overview of the solubility services which are available at Eurofins Discovery. So as I mentioned, solubility is the testing of compounds in various types of fluids. So we can also offer it in a simulated gastric fluid and an intestinal fluid, as well as fed and fasted formats of the same fluids. And we can use a number of different solutions, be they an aqueous solution or non-aqueous solutions. We also offer client-specific protocols. So if you have your own solubility assay, we can also develop that in-house at Eurofins. So as I mentioned previously, there are a number of methods for assessing solubility of your compounds. So these include HPLC, UV, and we can also look at them using a technique called nephelometry. And then on the left, you can see a table which gives you an idea of some of the solubility of some well-known compounds. So that solubility, I said, that's a very, very key parameter to be looking at for early discovery. And then if we move on, another key parameter is protein binding. So the extent of binding to proteins, predominantly plasma. So when we talk about protein binding, the most common protein looked at is plasma. So the extent of binding to proteins can significantly affect the therapeutic potential and the PK of a drug. So drugs bind mainly with plasma proteins, such as albumin, alpha-1 acid-glycoprotein, and other glycoproteins. So if you're looking at plasma, a drug exists in two forms within plasma. So either bound or unbound. This is illustrated in the diagram on the right. So depending on a drug's affinity for plasma protein, a proportion of the drug may become bound to plasma proteins while the remainder remains unbound. So it's important to note that the less bound a compound is, the more effectively it can cross cell membranes or diffuse across those membranes. If the protein binding is reversible, then a chemical equilibrium exists between the bound and unbound states. And this is called the protein-drug complex. So the protein plus the drug is equivalent to the protein-drug complex. So it is the unbound fraction which exhibits the pharmacological effects. And the bound form is usually pharmacologically inactive. So highly bound compounds have a lower pharmacologically effect than non-bound compounds. So protein binding has been shown to be a significant factor in a drug's biological half-life. So again, another really key parameter. So highly bound compounds, less pharmacologically active, essentially because they can't be metabolized, and because they can't, and also because it stops them from crossing the cell membranes. So again, there's a number of different ways to look at protein binding, and also a number of different matrices that you can look at. So the most commonly used method of protein binding is equilibrium dialysis. This allows you to determine both the bound and unbound fraction of a test compound. We can look at this using different concentrations of plasma. So if you have a very highly bound compound, you can look at it with a lower plasma concentration, and that will allow you to determine the fraction which is bound and unbound. As mentioned, it is predominantly performed in plasma, but we can also look at serum or albumin, and also we can look at things like brain binding and microsomal binding as well. So this should always be looked at in multiple species. So with the majority of ADME assays, as a first look, you would screen within a human species, and then also in one preclinical species, so which tends to be of a species from your pharmacology model. So if you're running a mouse in vivo efficacy model, you'd also look at plasma protein binding in mouse as well. So moving on from protein binding, you'll hear metabolism a lot. It's often, along with solubility, it's one of the first properties which are usually assessed in early ADME studies. So understanding a compound's metabolism is really vital in developing successful drugs, as obviously compounds which are rapidly metabolized may need to be dosed more frequently in order to maintain an optimal concentration, whilst those which slowly metabolize may result in toxic effects. It's interesting to note that many compounds with promising pharmacological characteristics never become drugs due to their poor metabolic stability profiles. So this is a really key area, one that chemists spend a lot of time optimizing. So understanding the susceptibility of compounds to undergo biotransformation in the body is absolutely critical for selecting drugs with favorable pharmacokinetic properties. So there's a range of metabolic stability assays, again, looking in different matrices and different species. And these measure the intrinsic clearance, or what we call CLINT, of a compound, providing critical data needed to calculate other key pharmacokinetic parameters, such as bioavailability and also half-life. The liver is the most important site of drug metabolism in the body, and this is often where you start first, is looking at liver microsomes. And approximately 60% of marketed compounds are cleared through the liver using CYP-mediated metabolism. So there are a number of in vitro test systems, as I mentioned. So these are liver microsomes. Those tend to be used at a very early stage, and then you can move on to hepatocytes as you move nearer to preclinical development. Also other fractions such as S9 and looking at blood and plasma. And again, similarly to the plasma protein binding, you'd look at both animal and human species, and this will provide you an accurate prediction of the stability of your compounds within various organs in the body. So again, we're looking at a combination of a number of key properties. So that's at the moment solubility, plasma protein binding, and metabolic stability. So this just gives you an idea of what a profile would look like of some stable and non-stable compounds within human hepatocytes. As you can see, propranolol is a very stable compound, especially when compared to compounds such as HFC. So again, just having a look at what's available at Eurofins from a metabolic stability assay standpoint, said we have a wide variety of in vitro test systems, including microsomes, hepatocytes, S9 fractions, and across a number of different species. So really focusing on the key parameters here, another vital area to look at is absorption and permeability. So oral administration is a preferred route for the majority of new compounds. Obviously, for some therapeutic indications, oral administration isn't possible, but for the majority of drugs, we're looking at an oral administration. With this in mind, it's therefore crucial that orally administered drugs are permeable through the intestine. Also, drugs which are used to treat diseases of the central nervous system are generally required to pass through the blood-brain barrier in order to achieve maximum effectiveness. So obviously there, they need to be permeable through the CNS. So the permeability is the ability of a compound to travel across membranes. So the membrane is effectively a barrier between a drug and its target. So there's a number of different in vitro ADME models which are available to accurately predict a compound's permeability, both intestinal and through the blood-brain barrier. So these come in two forms. So there's either artificial membranes, which are often used very early on as a cheap and efficient way to assess membrane permeability. So the most common is called PAMPA, which is the parallel artificial membrane permeation assay. And then also we can look at permeability across cell monolayers. Again, the most two common systems there are CACO2, which tend to be used to look at intestinal absorption, and then MDCK cells, which are used to look for absorption and permeability across the blood-brain barrier. So permeability for a compound is very much dependent upon lipophilicity, which is the final property which we will look at in the presentation, and then also pKa and pKb. Intestinal absorption for a compound really depends on permeability, solubility, and dissolution. And just quickly, if we have any chemists among us, some of the structural modification strategies to improve permeability include reducing the ionization ability, adding lipophilicity, reducing the polarity and the size. And also there's a strategy which is commonly used, and that's construction of a prodrug. So the table gives you an idea of some compounds which have a high permeability and some which have a low permeability. But all of these are marketed drugs, all with varying bioavailability. But it really is a key parameter. Compounds with low bioavailability, as we mentioned before, often need to undergo costly and time-consuming formulation strategies to ensure that sufficient drug is being absorbed within the body. If you can look at this early on and use modifications of the compound to try and increase the permeability, that's very much recommended. So again, just touching on what Eurofins offers from an in vitro permeability perspective, we have all the standard assays that you'd expect. So the CACO2, we have MDCK, and also PAMPA. Again, with all of our services, these are all customizable so that we can adopt your protocols within our laboratory. And then finally, the last of probably the 5 main assays to look at is lipophilicity. We've mentioned lipophilicity throughout the presentation. It's a key determinant of the pharmacokinetic behaviour of drugs. It can influence distribution into tissues and also absorptions and the binding characteristic of a drug, as well as being an important factor in determining the solubility of a compound. So as we talk about this final characteristic, I think it's clear to see that it is really a balance within ADME across all these different properties, that by changing the permeability of a compound, you may be affecting the lipophilicity. By changing another parameter, you may be changing the metabolic stability of a drug. So this really is a balancing act between all those different properties. So just to focus a little bit more on lipophilicity. So a compound with moderate lipophilicity has a good balance between both solubility and permeability and is optimal for oral absorption. As we mentioned earlier, the majority of NCEs now are looking for an oral administration. So also lipophilicity balances the cell membrane permeability in cell-based assays, is generally good for blood-brain barrier permeability and has low metabolic liability. If lipophilicity is too low, you'll have compounds which have a high solubility but have low permeability and metabolism. If lipophilicity is too high, you'll have compounds with a low solubility which are highly permeable and highly metabolized. So the main measure of lipophilicity is a technique called log-D. So that determines the partition of a compound between an organic solvent. We typically use octanol and an aqueous buffer. There are other methods, one of which uses HPLC UV. Again, just to give you an idea of the correlation of some log-D octanol values, and our services offer a high throughput log-D assay using a microplate with a technique called shake flask, and then also we have the more cost efficient and time efficient method of HPLC UV. So those are the 5 assays that people tend to use as an early ADME screen. So for early optimization, and then as we move on, there's also what we call the tier 2 ADME testing. So these are more intrinsic assays. This is where we start to look at the cytochrome P450 enzymes, start to look at transporter assessments and metabolites. So as I mentioned before, this is where we start to look at the regulatory requirements, what assays you need, what data you need to be able to submit a new drug application. So this is at the stage where you start to fully characterize your compounds, understanding that solubility here is really key. If you have insoluble compounds here, it's likely to affect the data that is generated from these assays. So these really start to prepare your compound for those preclinical and clinical studies. So our key assays for selecting, though, are your really top candidates. So for both preclinical animal studies and for clinical development. So the first parameter that some people tend to look at is CYP inhibition. And indeed, for some organisations that I've worked with, especially some of the large pharma, CYP inhibition, especially CYP384, can also be moved up into the earlier ADME assays. So I've worked with organisations where they look at CYP384 as one of the first assays, and if there's any liabilities, those compounds do not move any further within the discovery and development process. So just focusing again on CYP inhibition and phenotyping assays. So these look at both phase 1 and phase 2 metabolizing enzymes within the liver. And as we discussed earlier, these enzymes are responsible for the majority of drug metabolism that occurs within the body. So we're looking at drugs that interact with these enzymes by those which are metabolized by CYP enzymes, so that's CYP phenotyping, those which inhibit their enzymatic activity, so CYP inhibition, and also those which activate nuclear receptors, which lead to the induction of their gene expression. So Eurofins is able to offer CYP inhibition assays across all the major CYP enzymes. So that's 8 different isoforms that the FDA look for 3A4 in particular, which is a really key of the isoforms. You need to look at 2 different substrates for 3A4. So that tends to be testosterone and midazolam. And all of our assays use a recommended drug substrates as recommended by the FDA and by the CTA. You can use fluorescent substrates, but the gold standard is to look at these assays within a recombinant human CYP enzymes. And then for CYP phenotyping, that's looking at the stability of the test compound within each of the common human recombinant CYPs, we do that within a 96 well plate format. So that's CYP inhibition mentioned. It can either be a later stage assay, or some organisations do use this assay very much earlier within the ADME screening cascades. So CYP induction assays, another of the key regulatory requirements. So looking at drugs that induce the expression and activities of the CYP450 enzymes. This is usually done through activation of nuclear receptors and can dramatically alter the metabolism of other co-administered drugs. So you'll hear a lot of talk around drug-drug interactions. This is one of the key assessments for those drug-drug interactions. So those compounds which do induce the expression of these key CYP enzymes, that can lead to rapid metabolism and significant decreases in drug plasma concentrations, and also the loss of efficacy of co-administered drugs. So there are a number of regulatory requirements around the CYP induction assays. So the regulatory authorities now look for mRNA analysis. So by using real-time qPCR, and it's usually performed in cultured primary hepatocytes. So we can look for fresh human hepatocytes, or in more recent years, due to the improvement of some of the cryopreserving techniques, you can also use cryopreserved hepatocytes for CYP induction. CYP induction assays also give you the opportunity to investigate nuclear receptive activation of a number of key enzymes. So taking permeability and absorption, looking at these from a more intrinsic perspective, this is where we look at transporter, substrate and inhibition assays. So drug transporters are membrane proteins that are really involved in active uptake and the efflux of substances across the cellular membranes. So these are really known to affect the permeability of drugs. If a transporter is able to take a drug across a membrane. So transporters are known to be a rate limiting step in determining ADME, PK and toxicity profiles for drugs. Again, the assays available at Eurofins all comply to those determined by the regulatory authorities. So we can test whether or not a drug is a substrate or an inhibitor of a number of the recommended ABC and SLC transporters. Again, we can look at these in a variety of assay formats, including CACO2 cells, MDCK cells, and also HEC293, CHO, and membrane vesicles. So for metabolite profiling, there is specific guidance from the FDA. So on the safety testing of drug metabolites. So clinically relevant metabolites not detected in non-clinical studies, and also toxic metabolites formed in human versus animal species. So for metabolite profiling, for metabolite identification, you would look at this in human and also across a range of different animal species. Again, those tend to be the species from your efficacy models and also from your tox species. As you move further down towards preclinical development, you'll probably look to do this in a number of different matrices, including microsomes, hepatocytes, and also looking at plasma samples from your in vivo experiments. So towards the end of discovery, moving into preclinical development, it's recommended that you look at metabolite ID in a whole range of species. This is a really key parameter. If you're metabolizing your drug and it's forming toxic metabolites, these will also need to be assessed within the ADME assays, which we've touched upon here today. So now I've given you a very quick overview of key ADME assays. I'd also like to just walk through a screening cascade. So experience shows that different companies find different parameters more important. They may also identify liabilities of certain series of compounds early on and be looking to screen those liabilities out of the series. So, for example, I've worked with a company who had HERG testing as one of their primary screens because they noticed a HERG liability early on. So where HERG would normally be quite a late stage assay, they've moved that really early on within the tier 1 assays. We're just looking here, as we've mentioned, most organisations would start with a solubility assessment and also looking at microsomal stability. So looking at the metabolic stability within liver microsomes. So this is applicable for most targets. Then moving on from this early screening, you'd also look at hepatocyte stability and then those detailed metabolism studies, which we just touched on. So metabolite ID, the CYP450, inhibition, induction, and isoform identification. And then from here, most organisations, once they've started to optimise their compounds, will start to move a limited number of compounds within in vivo. So this is a common strategy for most targets. Then if we move across to the right-hand side, we start to see perhaps some more tailored strategies depending on the therapeutic area of interest. So as we mentioned, for CNS focused molecules, you'd look to do permeability with MDCK and DR1 cells. This will give you a good assessment of whether your drug can cross the blood-brain barrier. We look at plasma protein binding, and also specific to CNS, you could possibly look at some protein binding within brain homogenates as well. For those targets which are more peripheral, from a permeability perspective, you're more likely to look at a bidirectional CACO2 assay, in addition to plasma protein binding. And then finally, if we're looking at local and GI targets, from an absorption permeability perspective, you look at a PAMPA, you look at solubility and stability using a stimulated gastric fluid and stimulated intestinal fluid. And from a metabolic stability perspective, you'd also start to look at some gut S9 fractions. But then people use an in vitro ADME cascade to really prioritize the data which is most relevant to their drug discovery program. This also is a cost-effective way to look at ADME. So instead of screening your compound through all the assays at the start, you put a cascade in, and those compounds which don't pass your solubility and your microsomal stability criteria, you can go back and re-optimize before testing them in the more intrinsic and later stage assays. So an ADME screening cascade is very much tailored to your particular program, and our experts at Eurofins Discovery can help you to tailor and to design a screening cascade which is most suitable for your project. I won't spend too long on this slide. Obviously, we've spoken about ADME here. That's the main focus of the talk today. But also, it's key to understand there is other parameters which you need to look at. So those are in vitro toxicology and also in vitro safety pharmacology. All these parameters together, all these properties together, allow you to optimize your compounds to develop one which has the most chance of success as you move forward into the clinic. So just very quickly, just talk about some case studies. So to give you an idea of how some organisations really structure their screening cascades. So this was a very large project which we did for a large pharma organisation. So this was to drive their artificial intelligence platform. So they were looking to generate a significant amount of ADME data. So for this organisation, they chose to look at aqueous solubility by using nephelometry. Then they moved on to look at the drug-drug interactions. As I mentioned here, they looked at the CYP3A4, which really is one of the key CYP enzymes that needs to be investigated. Then they looked at absorption and permeability, metabolic stability, and also then log-D. So they built an artificial intelligence platform looking to be able to optimize the properties of their compound from an ADME perspective. Another large screening program for a large pharma was where we started to develop some custom panels. So here I think is a really good example of a screening cascade. So as you can see, they started off with aqueous solubility, and then with permeability and microsomal stability as those tier 1 assays, as the early assays. And from there, they prioritised which compounds had met with the key criteria within these assays. Then they move on to the more intrinsic assays, such as protein binding, looking at blood stability in both mouse and rat species, permeability, CYP inhibition, and metabolic stability. And again, another case study here. Again, we're looking at custom panels, and the organisation has seen many liabilities around the CYP3A4 enzyme. So they've brought that in again really early within the screening cascade, along with metabolic stability. So for this organisation, those are their key focuses of the CYP3A4 and the metabolic and the metabolic stability. So mentioned all these screening cascades are very much dependent on what the focus of your programs are. So just as we move into the summary, so hopefully I've given you a good overview of the tier 1 ADME assays and how they're used in drug discovery, understanding the importance of how soluble your compound is, if it's stable within a range of metabolic systems, how permeable it is, its lipophilicity, and also how it binds to proteins. All of these characteristics have to be balanced so that you can develop a high quality discovery compound to move forward into preclinical development. So our goal at Eurofins, and the goal of all drug discovery scientists, is really to discover candidates with very desirable PK properties, reduce drug attrition, and ultimately realise clinical success. So I hope you found the presentation of interest. I hope that you've learned something today about ADME and how it's used within drug discovery. So I'd just like to finally touch on just some of the services which we have at Eurofins Discovery. So we do have over 500 in vitro ADME and toxicology assays, that we've worked with thousands of customers within pharma, biotech and consumer health. We have a number of experts within our DMPK groups who'd be very happy to talk to you and to walk you through screening cascades, can advise on your data, how to move forward, which assays you should be looking at next. We offer very flexible custom solutions. So all of our ADME assays can be tailored or modified to suit your project needs. And obviously we have all these ADME assays, ADME capabilities, but we combine these with our safety pharmacology, our in vitro pharmacology, to really provide an end-to-end drug discovery solution. and then very